Hi. After reviewing all the details of the cell cycle, beginning with the interphase and then following the mitotic phase, the prophase, prometaphase, metaphase, anaphase, telophase, what we need to focus on now is what happens at the end of cell division. Simultaneously to telophase, there's going to be the division of the rest of the cell, the division of the cytoplasm, the organelles in the cytoplasm, and the plasma membrane. That is exactly what is explained by this concept of cytokinesis. There are going to be differences in how cytokinesis happens in animal cells and also in plant cells. And the reason for these differences is going to be largely depending on what is surrounding, what is covering the outside of the cell. In the case of animal cells, there's going to be only a soft and flexible plasma membrane. So dividing the animal cell, what it requires is these microfilaments. These are the same microfilaments we've studied before as part of the cytoskeleton of the cell. Remember also that the microfilaments are the thinnest of all of the cytoskeletal elements, but they also have this property of being contractile. Microfilaments, they are usually made of actin. They have the ability to contract and expand like a rubber band. And so what we'll see here happening, a band of these microfilaments just underneath the plasma membrane will begin the process of contracting, eventually inducing the cell membrane to cave in, forming the structure we call the cleavage furrow. The microfilaments continue to contract and contract until eventually it is going to split the cell into two. That is exactly the meaning of the word cleavage in this case. Cleaving something means simply cutting it into two parts. And the furrow is going to be this wrinkly appearance that you can see on an animal cell when the plasma membrane is caving inwards as a result of the contraction of all of these microfilaments. That is how animal cells are going to finish the division of the plasma membrane and the cytoplasms. Inside of the two cells, there's going to be a nucleus and organelles, and the other ones can have also a nucleus and organelles. Things are different with plant cells, because with plant cells, in addition to the plasma membrane, remember that there is also going to be a cell wall. And this cell wall is going to be hard, rigid. It is not possible to cleave a cell wall in a similar way you did cleave an animal cell. So what has to happen for a plant cell to finish dividing is that little vesicles containing the materials for a cell wall, specifically cellulose. Remember, cellulose is one of those polysaccharides. It's a structural polysaccharide. So these vesicles that have departed from the Golgi apparatus and now contain cellulose are going to begin lining up in the middle of the cell, right in the place that is separating one set of chromosomes and a nucleus that is forming on this side, and there's another set of chromosomes and another nucleus that is finishing forming over here. So right there between the two nuclei, these vesicles begin to line up, and eventually they will form a structure called the cell plate. Now, if you were to ask me what is the difference between the cell plate and the cell wall, is that we can refer to the cell plate as a cell wall that is still under construction. While these vesicles are still lining up, while there are still gaps between the two cytoplasm of the two cells that are about to be formed, we don't call this the cell wall. Complete cell walls will be forming later as a consequence of the fusing of the vesicles that were forming the cell plate. So now we can see that eventually plant cells will be divided. Each of the two cells will have its own cell wall that will separate it from the other cell they used to be connected to. And this is how cytokinesis, the division of the cell membrane, the cytoplasm, and the organelles of the cell will be accomplished in animal cells, and how that is different from the way it's done in plant cells. That is all for now.